Okay, so the other thing that I want to make a big point about is that I might be a scientist at BIOS, but I need a team. This is my current team, and they are sat there. So this is just a little bit of an example of what we're doing right now. So this is my love, which is Devil's Hole, the oxygen minimum zone. I won't be speaking about it today, but these are two of my favorite microbes, Chlorobi and Thaumarchiota. Then my technician, Jessica Godfrey, who does pretty much everything, but this is her favorite, marine fungi. My other technician, Charlotte Garish, she is looking at marine viruses. Then we have Cecilia Darder, who is an REU student that is research experience for undergraduate, and her project is looking at the sargassum microbiome and sargassum health. Then we have Catherine Pepin. She is a CABIOS intern, so she's from Canada, and she's looking at microplastics biofilm. And finally, Georgia Small, who is looking at nanoplastics, which are the really small plastics in the ocean, and she's looking at suspended nanoplastics. The plastics will be discussed later. Okay, so first of all, you can't talk about microbes without explaining what they are. They are single-celled, standalone organisms capable of living independent of tissues. And for our work, it's bacteria, archaea, protists, viruses, and fungi. So this is the phylogenetic tree of life. For the older people in the audience, there were two domains of life, single-celled organisms and multi-celled organisms, or bacteria and eukaryotes. However, in 1977, Carl Woese found a third domain of life, the archaea. And the reason for archaea being a third domain of life is they were linked to single-celled organisms, but they have a complex biochemistry and cell membranes. So they're more similar to eukaryotes. So they became their own domain of life in 1977. So why do I like archaea so much? Well, unlike bacteria, they have a cell wall that contains peptidoglycan. And eukaryotic cells, so us, may have started from two archaeal cells merging together. So the origins of life started with archaea. Now, archaea have a very strong cell wall. And I want to ask you all a question. How would this benefit archaea? Does anybody in the audience know? No? Protection, exactly. So archaea are known as extremophiles. They are capable of living in the deep ocean, which has a lot of pressure. They are found at hydrothermal vents. They are found pretty much everywhere where other organisms can't survive. And scientists believe that archaea will be the organism that is found in space. Okay, so bacteria can be autotrophic, so they are plants. They photosynthesize, or they can be heterotrophic, so they respire. However, microbes are a lot more complicated than that. And if you follow this flow chart, you can see that they can get energy from light, energy from inorganic oxidation. And there's some microbes that can breathe nitrate, carbon dioxide, sulfate. Okay, so I'm going to talk to you a bit about my microbial oceanography. And the reason I'm doing that is the main source of our funding, open ocean work. Do we need to do something? Oh.
1 second.
Apparently they just see me. They don't see the talk.
So I don't see a screen.
Do we move the computer? I was there.
Second?
Yeah.
Okay.
Oh, it's disappeared from here. Yeah. I mean, if I could look there. Okay. All right, we're back. Okay, so there's a few take homes that I want you to understand from the microbes in the ocean. Number one, the autotrophs in the ocean, or the phytoplankton. Their biomass makes up 0.2% of living matter on the Earth, and yet their photosynthetic activity makes up 45% of the Earth's oxygen. So every other breath that you breathe comes from the ocean. So I'm going to ask you, are our oceans important? Yes. Okay. So why are microbes important? Well, in a single drop of seawater, you get 10 million viruses and 1 million bacteria. So even though they're very, very small and microscopic and you can't see them, they are still important for the oceanic food web and for carbon cycling within the ocean. As I've just said, two pathways in the ocean are the biological pump and the microbial loop, and I'll explain these two to you. They are both extremely important to the ocean, and microbes recycle particulate organic matter and dissolved organic matter to higher trophic levels. So here we've got two schematics. The one on the left is the biological pump, and what it is explaining is the different areas of the ocean. So at the surface, we have the sunlight. So we have phytoplankton that likes the surface of the ocean, and they photosynthesize. They need the light, so that's why they're at the surface. When they die, they start to disappear down into the deep ocean. And as they're disappearing, bacteria are aggregating towards them and helping breaking them down as they go down into the deep ocean. So as it's going down, it's broken down smaller and smaller and smaller. This also is the same for zooplankton that eat the phytoplankton and then excrete fecal pellets that also fall down. Then we have the biological pump on the right side, which is basically saying the exact same thing, but I want to point out the microbial loop. And there's a particular important part of the microbial loop, which I'll explain in the next slide. But the biological pump is basically taking carbon from the surface of the ocean and sequestering it into the deep ocean. And if it wasn't for microbes, this carbon would be lost to higher trophic levels. So in this microbial loop, which is kind of fun because it's got a lot of cartoons, I just want to point out this arrow. It's the only arrow that's double arrowed. So microbes can use the dissolved organic carbon that is released when things are eaten, when things excrete. Because that carbon is in the ocean, it's dissolved. It would be lost if it wasn't for microbes taking that carbon up and bringing it back into the food web. And this is 40% of the carbon in the ocean. So as a result, microbes are very important. Okay, so carbon sequestration is a really big topic right now. Everybody wants to take the carbon dioxide and plunge it into the deep ocean. So the biological pump removes a huge amount. This is a petagram, or 10 to the 15 grams of carbon that the biological pump takes to the deep ocean. What happens to the carbon as it sinks to the deep ocean? Does anyone know? Yeah, it sinks, but so carbon compounds are a huge range of compounds. You have labile compounds, which are like sugars and amino acids that are easily taken up by organisms, or you can have recalcitrant DOM, or dissolved organic matter, which are the big aromatic ring compounds, and those are harder to break up. So as the carbon sinks to the deep water, it becomes more recalcitrant. It becomes the big compounds that the microbes aren't capable of breaking up. So the bigger compounds usually sink to the bottom of the ocean, whereas the labile ones are recycled within the surface of the ocean. Okay, scary slide number 1. 80% of the ocean is dark and unexplored, and the deep ocean is full of dead bodies and unknown creatures. Okay, so Bioscope is the project that funds my lab. So I need to talk a little bit about it. And our goals are to explore explicit connections to the biogeochemistry in the water. So we have a contour plot. Oceanographers love our contour plots. I will explain it to you. The x-axis is time, the y-axis is depth, and the color is what we're looking at. So in this plot, it's dissolved organic carbon, and the dissolved organic carbon is higher at the surface and lower at depth. Basically, what Bioscope is doing is we want to know what bacteria are there, who's active, what are they doing. We want to know what metabolic capabilities these microbes possess. So what are they eating? What are they excreting? What are they doing? And then we want to know what is the interplay between the physics, inorganic chemistry, and the microbes in the ocean. You know, what are they doing? Are certain microbes eating one thing and not another? Or do we have more of this microbe than that microbe? It's we are studying time series, so it's part of the BATS program, which is a very important program at BIOS. They go out on a monthly basis and sample the ocean to the south of Bermuda. It's very collaborative. We have about 7 different labs around the world, and we do a lot of experimental work on one of our process cruises. So I just want to explain a little bit more about oceanography as a whole. So again, contour plots with time on the x-axis, depth on the y-axis, and the color. And we've got 2 things. We've got the dissolved organic matter on the top. This is basically what heterotrophic bacteria eat. And then we got bacteria at the bottom. So all I want you to see is that we have a seasonal signal. We get the red color in the surface every year in the summer. Repeat, 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 repeat. In the winter, we have mixing, which redistributes the DOC throughout the water column. The surface DOC is typically labile sugars. The deep DOC is the recalcitrant aromatic rings. Now, the bacteria cells, we also get a seasonal signal. We get the winter mixing, redistributes the bacteria. It also brings up nutrients and DOC, so the bacteria can bloom in the spring, sorry, different way around. And then in the summer, they have a subsurface peak. And I want to ask you, why do bacteria have a subsurface peak? What could be at the surface that could cause bacteria to be less? Anybody? Sunlight, UV light kills bacteria. So that's why it's a subsurface peak. Okay. So these are just 2 studies that Bioscope have done. This is done by Shuting Liu at Keene University. She was at the University of California, Santa Barbara. And she wanted to know what types of bacteria show seasonality and spatial distributions. So at the surface, we have SAR11. It peaks in the summer, and it responds to the surface labile DOC. SAR11 is also known as Pelagibacter ubiqui, and one of our Bioscope scientists was able to name it because he found it. I'm showing the mixing. So this is the winter mixing when we have storms and they churn up the ocean. So that happens there. The second one, we have Flavobacter icei, which peaks as the mixing happens. So you can see this big red peak in the spring, responding immediately to the labile DOC that's brought up from the depth. And then finally, we have SAR202, which is low during mixing, and then it peaks below 150 meters. So what's really cool about SAR202, which has also been done by collaborators at OSU. They've looked into the metagenome, and they found that SARS-CoV-2 has the metabolic capability of breaking down these big aromatic ring compounds. So it has the genes for it, it's able to do it, and it thrives where that DOC is. Then what types of DOC show seasonality and spatial distributions? This is Krista Longnecker. She was formerly from Woods Hole Oceanographic Institute. She's been associated with the Bioscope project since it started. And she has shown that there is a summer signal, winter signal of pantothenic acid or vitamin B5. And this is repeatable. She's also found that tryptophan is high in July, and it's repeatable. Don't know why yet. We've just found this. Okay, I'm moving on to microbiomes. Does everyone know what a microbiome is? Yes. Okay, so this graphic is, I think it's pretty cool because they're showing the different types of bacteria groups across the top in colors. You can see plants have a similar microbiome, and then animals start to have differentiation of the microbiomes. And for instance, the pig, that's different sections of the pig. So microbiomes are different even within an animal. Now we all know the human microbiome, there's good bacteria and there are bad bacteria within the human microbiome. Here are a couple of studies that I thought were quite interesting. So on the left, we have all the different types of diseases that can be affected by an imbalance to your microbiome. And on the right, we have a study that looked at the gut microbiome changes with diet. So the early human diet had lots of microbes. Everything was happy and health was pretty good. Industrialized human diet, very low microbes. And they're mostly from Firmicutes and Bacteroidetes. Health status decreases. And then if you use microbe supplement diet, you get more microbes and your health increases. So you actually try to repair that imbalance that you have. Okay, scary slide number 2. There are more bacteria in the human body than human cells. So marine microbiomes, this is more what I'm familiar with. This is a paper written by Amy April. She's from Woods Hole Oceanographic Institute, and she's a collaborator, so excellent. The picture on the left basically shows the deep ocean, the different animals that thrive in the different ocean habitats. And if you think about all these animals, they all have a different microbiome. On the right is from the same paper, and she's just highlighting a few interesting microbiome relationships. So the bobtail squid has a bacteria called Vibrio fischeri. This is able to fluoresce and helps with its bioluminescence. Coral have a bacteria called Endozoicomonas, and this is useful for nutrient cycling. The killifish have tiny phytoplankton in their gills, which can help with oxygen. The humpback whale has 2 types of bacteria, and they're connected to health. I don't think Amy's figured out exactly why yet, but she knows that they're health related. Okay, so moving on to the sargassum microbiome. This is work that we actually do at BIOS. This is a study that looked at healthy versus unhealthy sargassum. So the offshore floating healthy sargassum had phototrophic and DOM degrading bacteria. It looks a lot healthier, it's happier, it's lighter. On the right, you have washed up on the beach, not so happy, brown, and it has degrading bacteria from the Vibrio species. This plot is basically showing the 3 different sargassum morphotypes. We have Natans 1, Natans 8, and Fluitans 3. And all I want you to take home from this plot is that the microbiome is similar for each one. And this is an ordination plot, and it's just plotting the different microbes that are found within the species of sargassum, and they have discovered that it's not necessarily by genotype of sargassum, but more where they are. So you see the Caribbean Sea has its own little circle, Florida, and then the rest clustered together. Okay, so this is a sargassum microbiome study we did at BIOS. This is the student that did it, Zoe Skinner. She was heading off to University of Surrey when she did this work, and she was looking at whether or not the bacteria on the microbiome were more abundant with one species or another. And we found a slight increase in fluitans compared to natans. And then we were looking at the sargassum microbiome. And again, it's very, very similar for natans and fluitans, with the exception that fluitans has a little bit more Alteramonas in it. So I want to talk a little bit about sargassum and the Sargasso Sea. So we have two pelagic sargassum, natans and fluitans. They live at the air-sea surface. They support 100 marine species, important juvenile fish habitat. They provide energy in the form of carbon. They're harvested to feed livestock. Cows are fed sargassum. Bermudians take the sargassum to use as fertilizer, and various other people do the same thing. And some people eat sargassum. But there's a paradox. The Sargasso Sea is characterized by nutrient-limited waters, and yet this sargassum thrives here. How does it do this? So there's a theory that could the sargassum microbiome break down naturally occurring phosphonates? And these are compounds that are naturally occurring or found in fertilizers. So they're actually brought into the sea using like the Amazon basin and the Mississippi basin. It's chemically stable. It's found in the oceans at about 70 nanomolar, and it can be degraded into methane and phosphate through the CP lyase gene. Okay, so this is the sargassum methylphosphonate study. Danielle Cox from the University of California, Santa Barbara, PhD student. She's just got her PhD. This was the first chapter of her PhD, and she's published the work. So on the bottom, I just want you to look at control bottoms with no sargassum and bottles with sargassum. Does anyone notice a difference? Yeah, the sargassum water is brown. That's all the carbon that it's producing. So we wanted to know, does the sargassum or its microbiome break down methylphosphonate into methane and phosphate? So what Danny did is she was measuring the methane that was produced in those bottles. So here's the plot. And all I want you to notice, can I do this? No, it's not coming up. So on the bottom, we have the yellow dots are the control. So it's a flat line. Nothing happened when it was just water. Then when it was sargassum and antibiotics to knock back the microbiome, it was a flat line as well. But when it was sargassum and water, no antibiotics, we got a lot of methane produced. And all those lines are just different experiments that we did. So the take home is that when you have sargassum plus the microbiome plus methylphosphonate, you get methane. You have sargassum without the microbiome and methylphosphonate, you don't get methane. So it is the microbiome that is breaking this bond. Okay, scary slide number 3. The Great Atlantic Sargassum Belt is 8.8 thousand kilometers long and weighs 2 million tons. The cleanup costs in the Caribbean are 29 million. Decomposing sargassum produces hydrogen sulfide, which is a health risk, and sargassum can smother coral reefs because the coral can't move. Okay, I'm moving on to drinking water. Amy wanted me to include some scary stats. So is bottled water safe? There's a couple of studies on the right that looked at bacteria and antibiotic susceptibility testing. So basically, does bottled water have bacteria, and is that bacteria antibiotic resistant? And they found resistance to 22 antibiotics in the bottled water. These studies were done in France and Portugal and Nepal. And the bottom one, if you can have a look, there's E. coli and Klebsiella, which are produced within the bottled water. Okay, so moving on to waterborne diseases. In the US, they cause 7.15 million illnesses, 118,000 hospitalizations, over 6,000 deaths, and 3.3 billion direct healthcare costs. What's in drinking water? Plastics now. So this is a study that looked at what types of plastic are found in bottled water, and polypropylene was the highest, and it was used in the bottle caps. So such a small thing can produce so much plastic. Another study found that in 1 liter of bottled water contained 240,000 pieces of plastic. So we're moving on to marine microplastics if you're not all scared. Yep.
Go back to the previous slide. I just want to, thank you. Thank you so much.
Okay, so marine microplastics. This is how marine microplastics get in the water, and it's basically from humans, industrial activities, and littering. As it gets into the water, it is fragmented due to UV light, oceans, and microbes. And then it gets ingested by fish, seabirds, aquatic life, and gets back into the human population. This is where oceans microplastics come from. And I was actually surprised that synthetic textiles was so high, and the plastic pellets, nurdles that we were all worried about a few years back is so low. So apparently we probably need to get all back to cotton. So this is what happens. So microplastics are bad enough, but you also have to think of the microplastic biofilm. So as microplastics are degraded in the ocean, you get a biofilm formation of microbes. Some of those microbes do include hydrocarbon degrading bacteria. And as they degrade the plastic, what are they degrading it to? So those are chemicals that are also being found in the sea. This is a study, and I really like this study because they used tar oceans. So they did, tar oceans was a huge study over all the oceans in the world. And it's a nice graphic that shows microplastics and then the biofilm forming on the microplastics. I really like the photograph over here, which is basically a microscope photograph, which shows the microplastics in light blue. And then those are all the different types of bacteria that are actually on the microplastics. So they've been stained different ways, and you can see them. So this is a study by Yoshida et al. In 2016. They discovered a bacteria called Ideonella sakiensis, which is able to break down plastics using 2 enzymes. So PETase and MHETase are the enzymes that it uses to break down plastics, and then other microorganisms degrade the products that they produce down further. Okay, so plastic colonization by marine microbes. It includes bacteria and larval microfauna. Multiple Pseudomonas species have been found on microplastics. Bacteria have been observed to colonize all known plastic polymers, and there's little understanding of bacterial colonization of various types of plastics over an extended period of time. This study looked at the microbial community of seawater compared to sediment, compared to microplastics. And again, we've got this little ordination plot and the little circles. So basically water is different from sediment, is different from microplastics. Other studies have started looking for antibiotic resistant genes on the biofilm of microplastics. And the reason they were doing this is because what they realized was that if the biofilm is on the microplastics, there's a lot of bacteria there, and they could be doing horizontal transfer of genes. So the more bacteria you have, the more bacteria growth, the more chance of gene transfer, and the more chance of antibiotic resistance. This study here was basically showing that you have microbial pathogens that could potentially come into the ocean. They could form on the microplastics. There would be hubs of pathogens with antibiotic resistant genes, horizontal transfer, and then these genes are getting everywhere. On the right, we have Catherine, who's in the audience, and her project, she chose to do this project. She wanted to see if the microplastic biofilm had antibiotic resistance. And I am not giving her results because she's still working on it. Okay, so scary statistics, global microplastics, only 9% of plastics are recycled, 73% of beach litter is plastic, and 171 trillion pieces of plastic are floating in the oceans. Moving on to nanoplastics. So nanoplastics are even smaller than microplastics. They're the 2 to 20 micron in size. You need a microscope and a stain called Nile red. And what Nile red does is it stains hydrophobic particles, and we use it to enumerate nanoplastics that are suspended in the seawater from 2 to 20 microns. And here's a microscope image of the microplastics, and you can see the filament. Okay, so this is a protocol that was developed by a student in my lab where we collect the samples, we stain them with a carpet dye, which creates a contrast on the microscope. We then filter up to 200 mils of sample through a filter, stain it with Nile red, mount it, and look at it under the microscope. This is the student, Madeline Regula, from Dartmouth University. She was presenting her work at the Biosymposium in 2023. And this is just some of her results, where we found more nanoplastics in the coast. We did Cooper's Island because it's a known microplastic hub. Inshore was lower, and then offshore even lower, which isn't surprising. You would expect that. We have done a transect from Bermuda to Puerto Rico in 2024. We're still working on the data. Maya Lighton, who is a Bermuda program student, she did the sampling in June this summer. And Georgia Small, who's also in the audience, is helping to process these samples right now. And there's only, we're still QCing the data, but we had more nanoplastics near Puerto Rico. And we actually found nanoplastics in the Puerto Rico Trench. Okay. This is probably the scariest slide that I'm going to show. Nanoplastics are in our blood. They can cross the placenta. They can be inhaled and have a potential link to asthma. The laundry creates 9 million microfibers. And nanoplastics can cross the blood-brain barrier and have a potential link to dementia. Okay, so we're winding up. Take homes. There are 3 domains of life. Archaea are extremophiles. The oceans remain largely unexplored. Ocean phytoplankton produce 45% of the world's oxygen. Bacteria in the oceans take up 40% of the dissolved organic carbon that would be lost to the oceanic food web. Microbiomes are important for health, and microplastics are everywhere. This is my team over the years. You might recognize some people. The kite is a microbe kite, in case you're not able to catch that. These are the funding sources. So most of our funding comes from the Simons Foundation International, which funds Bioscope. A lot of the sargassum work is being funded by a Cawthorne Innovation Award, which we got a new qPCR machine to look at that PHNJ gene. National Science Foundation supports the Bermuda Atlantic Time Series study, and we've also had support through local companies, Bermuda Hospitals Board, and Department of Health, and Helix. Does anyone have any questions?
you had a display of the different levels of problems that people encounter to do with this topic. But it didn't tell how many years was that. Was that 1 year, 2 years, 10 years, 20 years?
Oh, the
one
the
with-
carbon sinking down.
Yeah.
It's short term. It's not glacial.
No, it's the hospitalizations and the illnesses and
That's in a
deaths.
year. That's in a year.
It's in a year. Oh,
Yeah.
that's not good.
Yeah.
Hey, Rachel, you showed that slide of the microbiome on sargassum that distinguished between Florida and Caribbean Sea. What does your data tell you about the commonalities between what we find around our waters?
Haven't done the sequencing yet, Robbie. Yeah, I haven't. We're still, Zoe's going to be extracting a lot when she gets here. But we do need to send some off. I'm hoping, we're not quite sure if it's Jess that's going to go to UCSB in January to do the sequencing preparation.
This is a follow-up. You have other studies that look at the general microbial communities in the water between us and Puerto Rico. And anything sort of stand out in terms of if you're in Florida and everything sort of has to pass Puerto Rico.
I don't know about that. So one of the problems that I have with all the studies that are looking at sargassum is they don't really tell you where the sargassum is collected from. Is it healthy? Is it not healthy? So my theory is that the healthy microbiome is beneficial to the sargassum and has bacteria on it that can help with that cleavage of the PHNJ gene. Cece is doing a sargassum health experiment right now. She's got some results, but she hasn't got them all. I guess, could we say a little take home? So,
I guess part of my question is really, what's the commonality between what's growing on the sargassum versus what's in the water column within its normal?
oh, yes. So I think, so for instance, on the right-hand side, if people are studying the sargassum microbiome on the right-hand side, they're basically getting decomposing bacteria. So you're getting the proteobacteria, so you're getting the vibrios, you're getting the Alteramonases. So it's going to be completely different from the healthy side of things. I think that generally the microbiomes are similar, but there are some probably small changes depending on what microbes are in the water that can attach to the sargassum. We don't quite know that yet. I think the sargassum might, I don't know how it would do this, but I know corals do this. They actually take up bacteria that are beneficial to them. So I'm wondering if the sargassum do the same. In particular, Rhodobacteriaceae, which...
Where it hangs around long enough that this
Yeah.
sargassum bumps another piece of sargassum just keeps being
Yeah.
seared across the community. That could be meter by meter or kilometer by kilometer, hundreds of kilometers over time. So it's
Yeah.
cool that's
Yeah.
also being-
Yeah, so this plot might be driven by more of the rarer species.
Thanks.
Anybody else?
Thanks, you. That's an amazing amount of data and research there that you presented. Thank you very much. Way back, since you're moving back in the slides, when you talked about the seasonality of the bacteria, there seemed to be an anomalous year there, 2020, for some reason.
2020 had a
2002.
2002.
So it looks like there's not as much seasonality in that year. Bacterial cells. And I'm just reading that on the bottom.
Yeah, I would need to check that, but if you have a look at this color here, that's probably indicative of a deep mixing event. And 2010 definitely was a deep mixing event. It went down to 300 meters.
So in 2002, you just think that was a different weather pattern?
It could be that it was a shallow mixing and not enough nutrients, so the bacteria didn't necessarily respond.
Is that not at bats?
This is all at bats, yeah. all at the bat site, and I would need to talk to Rod about where the eddies were at that time to see whether or not. But I think that could be just a shallow mixing event that year.
I found something very similar in terms of microplastic distribution in water with my students' work. Inshore, across North Lagoons, up and down Ferry Reach, along the South Shore, quite elevated, very elevated sense from oxygen, from the historic stuff.
Yeah, and these are really small, so it's suspended in the water column. So we were expecting to see it more in the sites where you get more microplastics, of course. What we weren't expecting is to see it throughout the water column at bats. Anybody else?
The tests on the drinking water, what sort of drinking water was it? What
Bottled water.
Like just a particular brand, did you stick to the same brand?
I didn't do those
testing, or
studies.
testing? Was it done on one particular type of bottled water or?
So you probably get in a lot of trouble if you name the bottled water company in your study.
Or is it because of the plastic itself? Does that have something to do with the
Yeah.
plastic water, the fact that it's in a plastic water bottle?
Yeah, I mean, so you're getting the plastic from the plastic water bottle, and you're also getting bacteria. We have done it, and I don't drink Highland Spring. Highland Spring.
Highland Spring. Choose one of the names of others that you can drink.
No, Smart Water was the best.
Smart water.
Anything that filters is probably the best. Anything that has a filtration effect. If you're just going to some lovely spring in the Highlands, it's going to have bacteria associated with it because it's a natural environment. Anybody else?
It would be interesting to see how it compares to tank water.
So I have done that. Tank water will have stuff growing in it, but at home I have the clear water filtration system with the UV light, and it does work.
Yeah, but they say that the clear water, it doesn't, because it doesn't have any form of microbe, that it doesn't build up any resistances.
So I don't get that.
Like when you're drinking tank water, I mean, you're getting all different types of microorganisms, right? So to a certain extent, you'll build up resistances to separate. Whereas with the clear, the pure water, there's nothing in there. So all it's doing is actually hydrating you.
Yeah.
So
Yep. Well, that's why you let your children eat dirt.
or tank water.
Yeah, I mean, if somebody, quite often when we get visitors coming, they will get stomach upsets from tank water because they're not used to drinking it, whereas we've grown up with it, and you have the natural immunity. But the clear water system does work pretty well to get rid of the bacteria.
To get rid of it?
Yeah, I mean, you don't want certain bacteria. You don't want E. coli, and you don't want some of those other things. So, and putting bleach in your tank periodically does really help too.
I think that's right.
You do?
Yes.
It's just a very, very small amount of bleach.
No, for some people it's not.
So we have, I think our tank is 100,
Uh-huh.
and we put one of the really small Clorox bottles in twice a year.
I think they should send out a notice about that as to the actual concentrations.
Yeah, there is.
It's on the government site,
Yeah,
You have it?
4 ounces per thousand.
Yeah.
Yeah, I know they have it for restaurants and, for cleaning
It's on the government.
glasses and things, but a lot of people don't use that.
well, Desiree's daughter did an experiment for her IB where she looked at seawater and she treated it with UV light. She treated it with boiling. She filtered it, and she also used bleach. And bleach and filtration were the best.
That's wonderful news.
Mm-hmm. That was seawater. So, and there were some pretty. We even took a sample from forgotten the pond. No, Spittle Pond. Yeah. So we saw how much the bacteria changed from start to finish, which was cool. Does anyone?
The human microbiome, is it only in the gut?
No, you have skin microbiome, all sorts of different microbiomes.
Typically, when they speak about human microbiome.
Yeah, I mean, there's loads of studies being done on your gut microbiome now. But yes, I mean, that's human health. That's not me. There are lots of studies being done. I mean, you can Google, you can find out all this information.
And the gut would be from the mouth. to
Yep.
the head.
Yep. Anybody else? Have I scared you all?
Yeah.
Just before I say thank you to Rich, I'm just going to add a bit about the tanks and the tank water. A lot of it must depend on how clean you keep your roof, whether it's overhung by vegetation or not, and how clean your tank is. When I came to Bermuda over 50 years ago, I've never had a tummy upset. I've ever. Only recently put a filter on because of for chemicals rather than the natural stuff. But none of my visitors have ever had a tummy upset unless they've gone out drinking the night. So anyway, so I just had to throw that bit in on behalf of the beauty tanks and such like. But that was an absolutely fascinating talk. I just could not believe the amount of information that was contained there. It was. You will be able to find it on YouTube if you want to go back over it, because I'm going to need to go back over it. So it will be up. You will be able to find it. There's just so much information. Some of the scary facts, that about your clothes, synthetic clothes, putting all that out. I guess if you just shake it.
Yeah, it's scaring me.
Yeah, that is awful. I mean, but that
It's
is
also
the-
ropes and stuff.
Yeah,
Yeah, and all sorts of things that float
yeah.
around in the ocean.
Yeah. It's just so much information, so much more to discover. I mean, if we've only looked at 80%, 20% of the ocean, we've got so much more to discover. Big thank you to all the companies and institutions that support the research. Many thanks to the people who do the research, like Rachel, and then thank you for sharing it with all of us and the community. And there are lessons to be taken on board and what we can do about it. I did print off very small, because I didn't want to use a lot of paper, but that was also in the newspaper articles, what individuals could do to ameliorate the position or to increase your knowledge. So if you didn't get one and you want one, you can take one. But again, Rachel, that was fascinating. And I need to go and look at a lot of things up. Thank you so much.
Yeah, there's a lot
for
of
that.
information
Thank
out
you.
there.
That was amazing.
Thank you.
Thank you. And two sales pictures here. One, there's the library books outside. Please have a look at them and know that the Bermuda Library is open to the public. Bermuda College Library is open to the public. There is a subscription, isn't there?
Very small.
Yeah, a very small one, but it's not like the public library, but they do have a vast array of books that you can use, and a thank you to the librarian for doing that. And a thank you to the college anyway for supporting